Hi, how are you? This is Ahmad, and I'm going to talk about the epidemiological potential of excretion and regurgitation by Musca domestica in the dissemination of Escherichia coli into food. And this is my third presentation for this class. Author background Sasaski. He is a researcher in National Institute of Infectious Diseases in Medical Entomology Department in Japan. He has many publications with other researchers and he likes to work on Mustica domestica. Introduction 1982 Hemorrhagic colitis by Escherichia coli outbreak occurred in USA. 1996 other outbreak of EHEC in Japan. Although cattle are a reservoir of EHEC, the model of transmission has not been clarified fully for EHEC. In Japan, E. coli was isolated from both patients and houseflies. The researchers found cattle farms were located 30 meters from the nursery school. The collections of flies in the school showed that house flies and stable flies probably came from the cattle farms. The house fly is known to be an important vector of microorganisms such as Shigella vibrio and Staphylococcus. Recent studies showed that houseflies were not a simple mechanical vector of EHEC because the bacteria proliferated in the mouthparts and were excreted for at least 3 days after bacteria feeding, after which the number of bacteria decreased in the gut. Objective of this study. The objective is to clarify factors related to the rapid decrease in the number of bacteria in the alimentary canal and to discuss the potential of housefly in transferring EHEC. Material and method: housefly and Escherichia coli bacteria. Houseflies were used for feeding experiments. TSB media containing EHEC was ingested freely by houseflies for 30 minutes in a cage secured in safety cabinet for laboratory use. The houseflies were allowed to feed on milk or milk, water and lamp sugar for 6 to 8 days before use. Escherichia coli, two kind of or two E. coli or two strain of E. coli was used. Two strain of the bacteria isolated from Sakai, Japan, were grown in TSB at 36 Celsius for 18 hours. The concentration of EHEC was about 10 to the power 9 colony forming unit. Estimation of the number of bacteria. The sample was excised from house flies, anesthetized with CO2, placed into tube, homogenized with a plastic pistol and phosphate buffered saline, centrifuged to produce a clear supernatant. The number of EHEC in the sample was counted by plating aliquots of serially diluted supernatant on a on a EHEC, especially chrome agar plate. Colonies of EHEC appear violet on this plate. To reconfirm the colonies from the chrome agar plate by replating them on rainbow agar. Samples from control flies never showed EHEC positive result on a chrome agar plate. At 1, 3, and 24 hours after bacteria feeding, drops of excrement from house flies were collected with PBS 3 times. The solution was diluted and the serially diluted drop was applied to chrome agar plates. The plates were incubated at 37 Celsius overnight. That's the procedure. Effect of soybean trypsin inhibitor, which is STI, on EHEC growth. EHEC was cultured with gut extract supplemented with STI at 37 Celsius for 8 hours. The culture media were measured at 700 nanometer. Colony number was determined from a standard curve preliminary made during a growth experiment of EHEC. Estimation of the frequency of excretion. A small container filled with eosine supplemented TSP was placed over filter paper in a petri dish and contained one female or one male housefly per dish for 24 hours. After removal of the flies, red spots colored with eosine on the paper were counted, and the average number of spots excreted per fly was compared statistically. Result: EHEC in house fly samples after bacteria feeding. So for the result of this study, approximately 5 by 10 to the power 6 CFU of EHEC was detected in house flies immediately after bacteria feeding on both strains of EHEC. After 4 days of bacteria feeding, the number of both EHEC strains decreased to 25%. Effect of STI on EHEC growth. The number of EHEC increased from 5.7 multiplied by 10 to the power 8 to 7 multiplied by 10 to the power 8 CFU per mil in the presence of STI. EHEC in excreted drops. The number of EHEC in excreted drops was about 10 to the power 4 CFU per excreted drop 1 hour after feeding and 1.8 multiplied by 10 to the power 4 CFU per excreted drop 3 hours after feeding. The number of excreted bacteria decreased by 24 hours after feeding. Frequency of excretion in female and male house flies. The frequency of excretion by females with developing eggs and hair on their ovary was higher, 6.5 min per drop, than for males of females or females with mature eggs. Few of eosine sign around a container filled with eosine supplemented trypticase soy broth may or might be derived from frequent content by housefly, contaminated mouth parts. Discussion. Now, the transmission of pathogens by flies with contaminated legs or mouth parts are very important in transferring pathogens to human foods. EHEC were detected in the alimentary canal and excreted for 3 days after bacteria feeding. The number of EHEC in the crop of the housefly was greater and persisted longer than EHEC in the alimentary canal of the housefly. This may be because the volume of the crop is larger. STI significantly enhanced the growth of the bacteria when EHEC were incubated with midgut homogenate in a soybean trypsin inhibitor STI and grown in the broth. This means that trypsin in the alimentary canal of flies may be not the same because the compound studied significantly damaged EHEC. One reason for the rapid decrease in the number of EHEC in the alimentary canal is structural change in bacteria by the mouthpart of house flies. A mesh-like structure probably derived from EHEC completely packed the minute spaces of the pseudotrachea. Although the number of bacteria decreased after 24 hours, still the number of colonies disseminated by houseflies at 2 hours and 1 day after bacteria feeding. These results agree with Kobayashi study. The infective dose of EHEC is considered to be low. The data in this study showed that the reproductive age of the female strongly influenced the feeding behavior and the frequency of excretion. Females required nutritional material for ovarian development. The data showed that females with developing eggs fed and excreted more than females with mature eggs, and therefore had a greater potential to disseminate bacteria. It can be concluded that the house flies seem to be effective vectors of EHEC during the short period after feeding on bovine feces. And also this is my reference, which is the same article that I used for this presentation. Thank you so much, and please ask me if you have any question. Thank you.